Coming to principles of organ transplantation and immunological basis, these are multiple 3 mark questions asked. Coming to the steps, okay. So here it's not given clearly. I'll explain you like a step so you'll understand it better. So first is adequate exposure is needed. So first, when you are extracting an organ, what you do basically, first you give an incision, okay, first. So that's why by giving the incision, you expand it, no? So that's why more exposure is taken. Let's take for an example that you are extracting liver. So before taking liver, what you'll cut it and suddenly take it. So you have to control the blood vessels up and down. You have to clamp the blood vessels so that that necrosis and all will not happen. Basically, you are not affecting any other part of the body by extracting the liver. Okay. Once you cut the vessels, due to the body temperature only, the organ will undergo something called as warm ischemia itself. So that's why you control the temperature inside the body. When the organ is inside the body only, you control the temperature. So that is what is called initiation of preservation in situ. So just think of it as a inward cooling. The one is inward cooling and another one is outward cooling. Okay. So after you in situ preservation, you removal of the organs and separation. These and all are steps. Okay. First is organs separation and preservation. In some highly vascular organs, you do vascular reconstruction and then packing. Only the initial step are difficult. Okay, first is incision, then cutting the vessels, then in situ preservation, then remove the organ safely, and outside also you put it in cold. And what else? You preservation, packaging. That's all. Rest steps I said here. The same similar things are here. Like you make the organs viable and make them cold so that they can be kept outside also. So you inject inotropic drugs so that like the metabolism won't stop and there won't be necrosis. So, these are T3 and argipressin are used. This is in already brain death. In non-heart beating donors, some cardiac arrest, like you just think that you can write the stages. Okay, first is the person arrives, then you do resuscitation, which is unsuccessful. Then you wait for his cardiac arrest, and after his cardiac arrest with brain death, you extract. Okay, it's like 4 categories. All points are important. Whatever I say, if you remember, that is also enough. University of Wisconsin solution. So viscous means in cold there will be little stasis. No, it will become high viscosity. So that's why Wisconsin solution, like that you can remember. Donors organs are used for kidney transplantation. This you know, pakka. To immunological basis of transplantation, basically this CD4 cells and CD8 cells are mainly involved in rejection of grafts. Okay, average of these both 4 and 8. What is the average? So it is 6. So mainly HLA is located on chromosome 6. With this, you will never forget it. 2 types of rejections. This you would have read in pathology, but you need to remember that's all. Like what is the mediator? Okay, hyperacute rejection mediator is complement system. You know this because these preformed antibodies will be there. Now these are the main mediators of complement system. They directly whatever are there in the recipient, they already are there, they attack suddenly. So that's why hyperacute. Okay, acute rejection is lymphocyte. Mainly you have to remember that hyperacute, H for hyper, H for humoral. Mainly humoral response is hyperacute rejection. Acute rejection is lymphocytes, and even like acute and chronic, basically you have to remember that both humoral and cellular will be acting, but hyperacute or mainly humoral will be acting. After complement system, lymphocytes come in any like inflammatory response and all. So that's why acute rejection. Chronic rejection, fibrosis and scarring, and same like cell and humoral mediated immunity. So treatment, what do you do? Like you give immunosuppression, like anti-cancer drugs, like all sort of immunomodulators you can write. Okay, so it's just 3 markers. So you need to write like main the cyclosporin, tacrolimus, azathioprine, whatever are there. The headings you have to write. That's all. Okay, it's not that important. Like you can whatever I taught is more than enough.